This is NIME. So what this is my workflow, which segments nuclei and the cytoplasm away from cells. And the beauty of NIME is this has ImageJ integration. And if I enter into one of these nodes, we can see that I'm actually processing different things. So if I go back for a second, I can show you the input data. And I'll do this here. And so this is my input data. It looks black because the loots haven't been adjusted yet. But here I can do this. And hopefully you can see that a little bit. Increase that saturation. There we go. So this is my input data. And what we do is we track the nuclei as a means to know where the cell is. And the beauty with NIME is as you process these things, you can interrogate the process as you go. So for example, I'm going to go to this global threshold that I do on the data. So here's the result of that global threshold, right? Looks pretty good. And if I keep moving forward, I do an erode step, fill some holes. Here, I'll look at the label step here. And then here is my label step where I've done some cool things. So NIME gives you this power of combining all of ImageJ's amazingness, as well as NIME's cool data analysis things. It does like columns and tables and moving data around and manipulating it so that you can produce some really cool results. So I'll show you the output of this workflow, which looks like this. Yeah, I'll move this here. So right now what we're doing is we're tracking nuclei. I'm trying to zoom in. Here we go. Oops, too fast. And if I do time here, you can see there's a nucleus and then there's a cytoplasm. It looks pretty good. And if I go to the cytoplasm one here, you can see the trick we do here is we use rings. So we dilate the nucleus to push it out into the cytosol and then measure that as a proxy for the cytoplasm because segmenting cytoplasms is pretty hard. But yeah, so this is NIME. It's really cool. It has a variety of different functions. If I go over here and I go to the image analysis workflow, which is somewhere, it's at ImageJ. There it is. You get all these cool ImageJ functions. Yeah, there's a lot to talk about NIME, but check it out. And yeah, I don't really know what to say, but it's really cool.
Thank you. I'll definitely look into that.
Yeah, it does plugins too, so you can do plugins and things like that.
Yeah, Jan just asked if it runs macros still.
It does run macros. Yeah. There's a pure code. Oh, actually, do I still have this open? I do. I can show you real quick. If I go here, and here's an ImageJ macro node, and if I click configure, you can actually type in your code here, run, something, or you can do this pull down here and you can select some preformed functions. So let's say sharpen, and then, or here I did background subtraction. So there it is. So there's a background subtraction. And if I add sharpen, I can click sharpen and it's going to run that sharpen command. So I think you can also make your own commands and save them if you have something a little bit more complicated as well. But it does support, yeah, I'm sorry.
If I download NIME, does it automatically work with Fiji, or is there like
no,
more
what you're gonna have to do is you go to install new software and you'll type in image processing or something like that.
That's the package that I don't know what they call it, package or what they call it, but image processing is what you need to in order to access that stuff.
There's two. NIME has its own image processing package here, and you're gonna install that. And then there's another one, and this is the trick. You have to go to manage, and it's in the community extensions here because it's outside of the community, outside of NIME's community. You hit apply, and then you type in ImageJ, and then that will get you the beta version of the ImageJ integration that you need to get like ImageJ macro and all the other ImageJ nodes.
There's also integration with Cell Profiler, Elastic, a bunch of other open source toolkits. But not, I mean, ImageJ itself is not made for high throughput, like data processing. And that's why we partner with NIME, because that's what NIME was built for. NIME was originally built for like data processing and all this stuff. And so that's why we're integrated with them. So then you can really access, Go ahead, Ed.
Yeah, I was gonna say, it's really good. My only caveat with it is, as it's running through the pipeline, it stores that data. So you're going to end up using way more like memory than you think you will. So like, for example, on my 16 gigabyte laptop, I could handle maybe a 3 or 4 gigabyte, movie file, like a forward channel TIFF file, before I was starting to hit like the upper limits of my resources there. So that's something to think about. If you're dealing with like terabytes of data, I don't think KNIME's going to be your guy unless you can figure out how to do it in a way that it doesn't store that.
Look, we are working with like full slide images. I think maybe 1 gigabyte per slide, and we have like 20 of them.
Yeah, that's
fine. That's like the most.
Yeah, then you can process folders in that way. And if you got access to something that has like, I don't know, a terabyte of RAM or whatever, then just forget about it. And
QuPath might also be a good option.
That's what I was looking at. I don't know how to use
awesome. It's awesome.
Because I heard they had like the random trees pixel classifier or something, and I'm trying to get that to work. So I was wondering if they would be here today, because I tried using it because we're trying to analyze ADIPOcytes, and ImageJ doesn't do the whole slide image analysis. So we're trying to use QuPath to see if we can do through that. But
so QuPath, they are active on the forum. So like Peter Bankhead always is answering questions, and there's a couple other power users of QuPath also that answer questions on the forum. So if you have a specific question for QuPath, definitely go there because you will get an answer for sure.
Okay, I'll try it out again. Thank you. This was all like super helpful.
Yeah, no problem.
Good.
Oh, I have if you want to get that KNIME workflow, let me post a link to it. And you can actually just use it as like a starting point if you want to try it and just chop it up. So just a second.
They also have a bunch of built-in examples too, I think, that are in part of that image processing file or package or whatever. They have like template scripts or template workflows that you can start off of. So you don't have to just start from scratch.
There, you can get the workflow there. And my old lab name it is HIV lab, but just ignore that part. There you go. So if you actually import that, and I think there's a tutorial there as well on how to set up KNIME with ImageJ. And if you just follow that tutorial, that should be okay. It's in the doc folder.
I might add for very big images. I mean, ImageJ is definitely, potentially not made for that, but in some situations, at least you will run into trouble. But for example, the CLI-J2 library, which runs on a graphics card, has functions, and there are new ones coming in where you can chunk the image into pieces and basically only like push pieces to the graphics card and then step by step run your procedure on that pieces, and later on just puzzle your image back together. So obviously it might take longer, but if you have a good computer, good graphics card, then it might be actually also for, especially for slide scans or so, which are bigger, quite good. But I think I would also corroborate that especially if it's histology or so, then QuPath is the way to go.
Are there any other questions? And if you don't want to talk, you can also add them to the chat. That's totally fine. So even if they're general questions or anything, don't worry about it. Are there a bunch of people here who tried doing the segmentation of all these data sets? Did you guys take part in the challenge and submit?
I took place in the challenge, but I didn't submit anything.
Oh, okay.
I looked at the challenge and I was like, I have no idea how to do any of this. So I just.
That's what I said. I was like, well,
I know, right?
And then Ellen told me about the second set of nuclei. I was like, oh no, my first results are terrible.
I know, like it was so funny because we were like, oh, we got this, because we both were just like looking at the first data set and we're like, we got this, and we're like, we got it, it's done. And then all of a sudden we were like, no, it doesn't work for all the data sets. So we were
learned
like,
my lesson. Check all your data sets before you start.
Yes. Well, most importantly, acquire good data right from the beginning that can be pooled quite easily. So that's the biggest takeaway, in my opinion.
Yeah, the histogram is really powerful. When I was a scientist, I wasn't using it enough. I guess I'm still a scientist, but I wasn't using it enough. And now I know how to use it. It really gives you a nice overview of what's the data is like a little bit. So definitely.
Well, and I would say, like, the most important thing is too, if you're doing a large scale image, if you're planning a large scale image analysis workflow, do pilot experiments. first. That is like that's the one thing I had to learn, because I was a microbiologist. I studied gastrointestinal pathogens. So we were using animal models. And in our case, we had to do pilots first, just because you can't do a large scale, you can't just start with a large scale experiment and, use up a bunch of lives and throw your data away. But I would say that's the biggest lesson I learned, was do pilot experiments for your image analysis to make sure that you're acquiring good data and that you're getting the good data out at the end, and then you can scale it up. I would say that would be my biggest tip.
I would definitely second that. I'm working with my previous lab and they want to segment some things and they didn't add a nuclei tracker or stain. So I'm like, well, can't really do anything with that because my workflow requires a stain. And they have to recreate all the data, which is unfortunate because they had the space, the spectrum space for it, but they just didn't add it in there because they weren't thinking about trying to analyze the data at that point. So definitely just think about, okay, what am I tracking on? What other markers I need, and make sure that they're of sufficient signal to noise ratio.
And that you include all their controls.
Yes.
in a single experiment. You don't want to
Yes.
analyze your controls separately from,
No, you don't.
so stuff like that. It's hard because I think also many times you get biologists who come and ask you for help for their segmentation. You're like, well, yeah, you can't answer that question with that data set. And that's heartbreaking because that's time and energy that was essentially wasted. It's not wasted because you always learn something along the way, but yeah, so just something to keep in mind, that you always plan those things well in advance. And that's what the forum is good for too. If you have questions like, this is what I'm planning on, or this is my pilot experiment, how does my data look? Can I answer this question? That's what we're there for, right? So definitely head to the forum. I'll link it again here in the chat.
Okay, I'm going to stop recording because the potentially exists that we'll get booted off at 1 o'clock. So thank you.